Today I'm going to be talking about the Inferior olive's digital twin, a large-scale computational model of the inferior olive. Its departure point is the idea that you can take experimental data into a computer model and make predictions with it. So we made a biophysically plausible computational model of the inferior olive in large scale. This is in the context of the project for the Horizon 2020 within the Human Brain Project. Arborio, which is supposed to integrate a detailed model of the entire murine inferior olive with a large-scale cerebellum model from Pavia, Egidio D'Angelo, as a compelling use case for the novel simulation language Arbor. And we are endorsing Arbor the same way as we're endorsing our own Eden in the context of the computational challenges that we face when we're doing a very large-scale model like this one. So I emboldened some of the key challenges here. I'm not going to go through each of them, but I'm just going to point out that the inferior olive has a dense gap junction connectivity, as you just heard from Georgios Smaragdos a week ago. Then we have the problems of parameter combinatorics. So it's really astronomical, the kinds of parameter spaces we have to traverse, and we have to try to think about those parameter spaces also biologically. So how is it neurons adapt to adjust themselves with the dynamics of their neighboring neurons. And we wanted to model the local field potentials. This is going to be in the bottom slides. Eventually to simulate a human-sized olivocerebellar complex with Egidio. We are at the point now that we've finished our olive, or pretty much, and we're going to be able to hook it up with Egidio. So what I'm going to show you now is our preliminary scientific data. The work that is involved here is we start from single-cell morphological reconstructions. We port and tune them. We place the neurons in the networks. In fact, in the sheets that the nucleus seems to have, according to reconstructions. We validate the anatomical data. I'll show you that. We have developed, together with Leonard Lensmeyer, who probably is here, the master student that has been driving this project. We have a set of auto-tuners for the network cells so that they adjust themselves to believable dynamics. And then we look into network parameter spaces, do some model validations via electrophysiological metrics, whereupon we're finally able to do some science, that is predictions, hopefully. Let us begin. You might have heard that we have these inferior olive compartmental cable cell descriptions in my past talks. The model we had previously was a simplified three-compartmental model simply with a soma, an axon, and a single dendrite, so only 3 compartments. But now we have an entire set of reconstructions from which we can learn how morphology influences the dynamics of individual cells. So these cells, given their ionic mechanisms, as you have here, so potassium, sodium, like the usual suspects, calcium low thresholds, potassiums, calcium activated potassium channels, their interaction produces the 3 different behaviors that you see here in the bottom left: non-oscillating, subthreshold oscillations, or even spiking. We have these absolutely gorgeous reconstructions to work with that we've been using so this particular tracing has been produced by Nora Freeler, who is currently visiting us in Alexandra Badura's lab. The render itself is in Blender by Leonard, the master student whose name you're going to hear often today. We take these reconstructions and we make them computational models by ascribing the channels on them. We are using NeuroML, so neural markup language, to describe these cells and networks so that we have ultimate portability and human readability, so that we can run these cells in different simulators actually ultimately as well, and have a more modular description of the cells that is easier to change, or indeed is easier for anyone to work with. This is a screenshot of the open source brain site where we have our inferior olive. If you want, you can play with them. You see how easy it is. It should be just a matter of clicking around to see what is the behavior of the ion channels that we have in the cells. And so I just wanted to tell you a little bit about this attempt to, make our models as available as possible and as usable as possible, as useful, and in fact, straightforward for even a beginner to use. If you're curious, you can ask me. Then we take these cells and we make. I've been keen to try to reconstruct an olive that looks like an olive, because there are certain anatomical features of the olive that are really fundamental for its operation, particularly the fact that some of the nuclei are organized in terms of what would look like sheets, so almost like cortices inside the brain. So we define a boundary volume from a selected 3D mesh file that has been reconstructed by a very competent neuroanatomist. I'm going to show you in a moment. We positioned some potential points where the cell somata will be, and some other distribute uniformly some points for the cells to grow within this volume, if you like, so as to avoid each other. So we want the dendritic trees that we're growing. So this is synthetically grown dendritic trees inside this volume to avoid each other, but nevertheless meet in the so-called glomerulus, which is the place where gap junctions, most gap junctions happen in the inferior neurons. So basically, we take this volumetric reconstruction that Tom has produced with these surfaces, and we fill them in with neurons. I wanted to tell you about the difficulties that are lurking in here. So for example, in the principal olive, you see, like, if you look at my video for a moment, so the principal olive is here. And you can see that it's folded inside. So to insert cells, making sure that the dendrites don't cross the fold is a pretty hard problem. But it's also a pretty fundamental problem, because even in the human olive, we seem to have the inferior olive organized as a sheet. So this is the principal olive in one of the very beautiful micrograph here from Economo, early 20th century. Actually, he was born in end of 19th century. Greatest anatomist there. So we, with our algorithms, were able to fill up these networks, and so these are synthetic neurons that grow within these boundary representations. In different colors here are simply different neurons for you to be able to tell how they look like. In gray, you'd see some other neurons. I mean, it's just to give you a sense of their morphology. But the kind of networks that we're now able to run are really dense. And here on the bottom right, you have our PO filled up with neurons. Now, this is not enough. So we have the reconstructions from Nora, and we have computed a bunch of metrics in this from these cells. And we're trying to make sure that to the extent of the possible, our metrics match with the distributions that we find in reality. There are a couple of like substantially different metrics here. For example, number of branches and number of stems. That's due to our assumptions on how to build these networks. But importantly, the dendritic total length and longest path and straightness and reach and number of connections. Those are the electrophysiologically most relevant variables, and those have a substantial match. The number of branches in the experiment is much higher, but tend to be relatively short. Now, it's important that we have electrotonically plausible cells. So then we go from these reconstructed morphologies to networks of cells whose dynamics are plausible. We use evolutionary search to do so. This is a method that I'm not going to go very deep into, but I just want to tell you that we're first basically finding a subspace for which the cells respond passively similarly. And the problem that we have in general here to optimize cells is that it is really hard to optimize every single one of the cells. So we basically try to sample cells in terms of their morphological similarity and attribute particular dynamics as a function of their shared properties. So we would then measure the impulse response to a couple of different perturbations in the soma and in the dendrite, and that would give us a relatively well-formed subspace for those electrophysiological properties. We then look into the different, how the evolutionary search produces believable cells. Here you have scaling factors on the left side here with the 5 best individuals for 2, 3, or 4 main dendrites in the cells. And we can interpret this data is a very interesting point, but I don't want to go there right now. What I want to tell you instead is that this evolutionary procedure gives us a confusing set that is repeating and recapitulating biology in terms of cells that oscillate, cells that are actually dampened oscillators, some cells that can be driven to oscillate, and some, in the case of a gap junction network, what we really truly have is driven, dampened, coupled oscillators. So the proportion of these cells that we are putting in our network has been obtained in experiment, and we have to mix them all together to find like I mentioned before in the beginning, we have a humongous parameter space. We have to mix them so that they talk to each other and that they keep their behavior both in the case of gap junction network as a non-gap junction network. The kind of problem that we have is that if you remove the gap junctions from a modeling network like this, you have the problem that you have increased leak, and that kills most of the trained cells from the evolutionary search. So what we had to do was to understand our parameter spaces, basically how the combinations of parameters influence the oscillatory properties, obtain those parameter spaces from a vast class of model cells, and really give the cells the ability to change themselves when we decouple the cells from a gap junction network where the cells have been evolved to a connexin 36 mutant, like a version of the model that has no gap junctions. We have to compensate to find the same distributions of oscillatory properties, and we do that via a auto-tune method here that I just wanted to quickly symbolize in the bottom left, or actually the x-axis here is the gap junction conductance, 0, 0.001, and 0.01, meaning millisiemens per centimeter square in terms of conductance. If we don't, this is just to show that our control system works. It might be too faint for you guys to see, but if you can see my mouse here, there's a dashed line. This dashed line is the level of expression that the neuron has selected to have in order to have the oscillations at the right amplitude, and this is as the gap junction conductance changes across the cell. So we're able to keep largely similar behavior across different gap junctions. Now, I have 5 minutes to show you this. This is the model. So now the original model we had was just somata, but now we have the activity of these individual oscillators in these reconstructed networks with dendrites. In this case, we're showing the principal olive. if you analyze these dynamics, and I'll show you a bit more data later, you'll see how these networks participate together in the oscillation. This is the principal olive reconstructed. And thank you, Leonard, for rendering this and actually writing the custom shaders in OpenGL so that we could actually show the activity in the dendrites. Now, if you ever heard any of my other talks, you would know that there is evidence for anatomical clustering in the inferior olive. From the left is a paper by also the paper by Nora and Mariuka and I, and on the right is a dye injection by Tiho, showing that the cells like to come in bundles, but these cells are not coming in isolated bundles. The clusters are most likely connected to cells further away, and that's why Mariuka and Nora decided to call these cells the curlies and the bridges, right? So the clusters and the bridge cells. So you have local and global connectivity. This is a hallmark of what is called a small world connectivity, and it leads us to believe that these clusters will actually organize themselves also dynamically. Clusters in different parts of the olive will come to oscillate together in different times. We want to look into this and analyze this so that we can have some telltales of the experiment. This is just to show you that we can parameterize our network construction methods to create clusters. This is a cluster. It is JAG because these are compartmental neurons. This does not have like a direct impact on its electrophysiological reconstruction of the model. Once we look into the oscillations that the cells of the model produce, so you saw those flashing lights. Now we are looking into different model cells here, and you see that they produce a very rich, complex set of oscillations. Nora sees this in experiment, and she calls them the wacky oscillators. Now we want to understand how these wacky oscillators oscillate together, whether in the same groups or different groups. We do this via dynamical clustering analysis. The method is relatively simple. So for each time that we want to see if there's a dynamical cluster, we find the next 10 oscillation peaks. We find regions where the pattern of peaks between two pairs of cells is interleaving. Because if there's two, then there cannot two oscillations of one cell between the two oscillations of the other, then they cannot be phase locking. They can only be phase locking if there's 1-to-1. We calculate the phase differences. We perform linear regression through the peaks, only keep the ones with a slope z-score below 2, and then we extract the community structure, that is, which are cells that are most likely together via a Louvain algorithm, whose objective is modularity. Here you have 10 seconds of behavior of 10 neurons, and the bars indicate when two neurons were oscillating together. So neuron 2 it was oscillating with, see maybe this top color here, was a partner of a neuron for a long time. This looks like they're most likely in the same cluster. Some other neurons participate in the same pairs for a shorter time. In the middle here, I actually had a stimulus. So this changes the allegiance of the different neurons, demonstrating dynamical clustering. So we can also look into this as how the cells are together in the same cluster over time. So here we have, you can see the 1 second bar there. This is basically which cells are belonging to which groups at which times. If you cannot understand this, I am more than happy to talk to you about this very soon in the future. I have 2 more slides for, because they're exciting, and then I'll close up. We do knowing from the model that we have these different cluster communities. What we can try to look now is whether there are counterparts in the experimentally measurable outcomes that we can look for, right? So look for the telltale signs of this type of dynamical organization. We do this by reconstructing the local field potentials. This is a book just coming out from us in Springer. It's called Computational Modeling of the Brain that we're edited. There's this beautiful chapter by Galtani-Fold and collaborators on how to compute the local field potential. And here, so here we have on the left a spontaneous field potential from a slice of the principal olive in our case, and you see some community structure, some clusters oscillating together. If you now produce Poisson stimulus onto those networks, that community structure disappears. I'm going to jump a couple of slides in the future. So this was to give you a sense of the fact that we can now, via the local field potential, understand what inputs will do to the community structures. This is a set of clusters that we can retrieve from this network. And my last data slide is this, in which we show that what happens if you give GABA during these gray areas, Poisson GABA, what happens for the local field potential above as an average and below for the V soma. You see that you just measure for the membrane potential of the soma. You see that in the membrane potential of the soma is a much less complex signal, but there's a lot of what we can read out of the number of components in the oscillation in the slide above, in the plot above. So even though it looks complex because it's local field potential and no one understands it, we can extract the number of clusters from this, and this we have confidence. To wrap my talk, I'm 3 minutes over. The IO's digital twin is an anatomically and physiologically detailed model. Thanks to Tom, Nora, Yoe, and others. We do synthetic generation of neuroanatomy with wiring and clustering as a function of known experimental data. We tune the cells via evolutionary research with communities, and we tune the networks with auto-tune of individual cells. We do statistical validations through spikes and LFPs. We look into the dynamical clustering, and all of this, you have, anyone of you that now wants it, has it in a big Jupyter notebook that Leonard prepared for you. So this is the Arborio team. Thank you for everybody, but in particular, I mean, none of this would have been possible if I wouldn't nerd snipe Leonard Lensmeyer with ideas, and he would come up after the weekend with the solution. So I am really happy to be very thankful to one of my most extraordinary master students I've ever had. I am happy to take questions now.
Thank you very much, Mario. Well, maybe I should start with asking a question. I saw also, of course, how GABA was implemented in one of these simulations. And did you there also, or did they implement the shunting mechanism?
Yes, we have a shunting mechanism in the glomerulus. So okay, it's via the tau, yeah.
So it's the same effects as described, let's say, by Linus and Lang and others.
For sure. Also, so if you have GABA in the dendrite, then generally you will actually shunt the dendrites and impede communication, just as also Mariuka showed in her paper with Yara Leffler. So a very easy to reproduce phenomenon, actually.
Okay. So you get a kind of leakage current.
You do.
Yeah. And how did you deal with then with the excitatory input? From the
so for the moment, we haven't actually started testing the excitatory inputs yet. We have them there, but we are now, one of the important things that we have to reproduce is the connection between the cerebellum of Egidio and our olive. So what we're doing is basically we're going to Pavia with the detachment of ours, so that we decide on how the arborizations coming into the olive look like. So we know we're going to use the same type of AMPA that we had in my previous model, potentially adding NMDAs. And but for the moment, we haven't actually done the research yet. They're there, ready to receive stimulus. It's basically the same as we had before.
Okay, so it will be implemented eventually. Yeah.
It is implemented. Now the question is, what's the scientific question that will use it? We have the types of experiments that we've done in the old model for Tycho Hoogland's and Sebastian Loyola's paper are easy to reproduce. Let me just share a thought here quick.
Well, of course, I'm going to ask about the timing between the two, right?
Yeah, right, exactly.
Yes.
So those resonances, I think that we can also try to look into. Now, this is, and to validate our previous models. As we go forward, we want to, be doubly constrained through experiment and through earlier modeling. We want to see if they match. If they don't match, why they don't. So far, it seems that the extra morphology hasn't invalidated our observations so far. So we will continue to look into this, and I am excited to now look into the comparison between the local field potential data here with the local field potential data that we had measured before directly in the olive. You remember the experiments with the ventral approach in which we had the electrodes inside?
Yeah.
So we still have that data. And so we want to, that's our primary goal, to reproduce the data from air puff into the whiskers as the mouse is anesthetized, but receiving, but being recorded at different depths in the inferior olive. Yeah. And so we want to look into the LFPs there.
Okay, I want to give the floor to Hugo.
Yes, thank you. So your talk was mostly about the model itself, but I just had a hopefully quick question about the simulator. So yeah, what are the strengths and weaknesses of Arbor compared to other simulators?
Oh, I hope that Leonard is here. So Leonard, are you here? I can definitely answer that question. But yeah, so this is my master's student. He has been grinding and hacking away, so I think he's much better equipped. He is a believer.
I am a believer. I don't have much experience with
stop sharing, Mario, so everybody can see Leonard.
Sure.
But I think that the main good point of Arbor is its speed. It's in experiments about 100 times faster than Neuron in most cases. And the nice thing about Arbor is that it's delivered as a library. So you can either plug in your model directly, or you can just go a little bit low level and start writing coding at lower levels. For example, if you want to implement a control mechanism inside your cells, I could only find it possible inside Arbor until now.
So Arbor is flexible and fast. I mean, it has a couple of disadvantages as well. Would you like to add to the disadvantages? Else I will. Yeah, so flexibility, comes a lot of freedom comes with responsibility. So other programming languages try to constrain you more into their, and other simulators try to constrain you more, especially, for instance, like descriptive languages like NeurML, although Arbor is implementing that. I feel if a noob goes into Arbor, I wouldn't be surprised if he committed some blunders with respect to, say, for example, scales and units, physical units. I mean, there too much flexibility makes you forget that you're dealing with, picosiemens, nanoamperes, and so forth and so on. But that wouldn't happen ever with Leonard. That would just happen with a newbie.
Okay, then the final question is for Wouter.
Okay, hello, good morning, Mario. Thanks for a great talk. I was intrigued by slide number 20, where you say basically that the cells or the concatenation of cells can behave like oscillators, damped oscillators, driven oscillators, driven damped coupled oscillators. And then I was thinking of something that happened to me in the field of electronics, that these type of oscillators sometimes can also show chaotic behavior. And I mean chaotic in a mathematical sense.
Dynamical systems guys, so yeah.
Yeah, so does that play a role, or why isn't it included here then?
Yeah, it is included naturally, just because the oscillators are Yeah, they're chaotic. So the inferior cells are chaotic. Now, there are two reasons why I don't emphasize that point. One is that the stochastic input overrules most of the chaotic properties of these networks. I mean, so they operate as low-pass filters often, and even though chaos is about sensitivity to initial conditions, takes a long time to express itself. So you would only see the divergence in the trajectories over a very long time. But if you have common-driven input from the sensory feedback system or from the cortex, I mean, often what you're doing is you're driving those networks. And I think that the most important feature of this set of oscillators is to absorb the resonances of the neural system to then produce appropriate cycles for the brain, so to speak. So it's asynchronous, but driven. And so these oscillators, they definitely resonate. If you bang them, they will lovely synchronize for a little while. In fact, there's, let me show you one more slide, because the what I had here, and I think it's hidden, unfortunately. If you allow me, I'm just going to show you one of the hidden slides. Did I delete? Oh, no, here. So the entropy of the system is interesting. If we give, and say, for example, in this case, an AMPA stimulus, as Chris asked me, what happens with the community structure? The first phenomenon is what we observe also in experiment is a large-scale synchronization that puts all of the groups together. Reduces the complexity of the system. The entropy is gonna drop down to 0 because depending on the amplitude of the stimulus. And then just think about this. So the cerebellum and the olivocerebellum is for motion. How long is the memory that we need for a gesture? We are operating on a temporal scale that's 100 milliseconds. It has to be on the fly. The transients of the neural system, especially the olivocerebellar system, are super short, 300, 400 milliseconds at most. So chaos doesn't matter there. Stochasticity matters a lot.
Clear, yeah. Thanks a lot, Mario. Excellent.
My pleasure. Thanks for the question.
Okay, guys. I think time is up. I would like to thank everybody and thank both speakers once more.
Thank you, Leonard. Thank you, everybody.
Good work.